for you all to read upon request in the library. We're thrilled to have you here tonight for what promises to be a thought-provoking and possibly paradigm-shifting conversation. Shortly, you will hear from Dr. Gerald Pollack. Dr. Pollack is a professor of bioengineering at the University of Washington and one of the foremost experts in the field of water science. He is the founding editor-in-chief of the journal Water and the author of several books, including Cells, Gels, and the Engines of Life, and the Fourth Phase of Water. You see, if Dr. Pollack's theories are correct, then everything you learned in biology is wrong. That kind of challenge isn't just controversial, it's revolutionary. And whether or not you agree with his theories or think they're correct, the questions he raises are worth our attention. So tonight, I encourage you all to listen with open ears and an open mind, think critically, and challenge perceived perspectives. Without further ado, please help me introduce Dr. Gerald Pollack.
Well, thank you. Thank you so much for the introduction. Can you hear me? Okay, well, that's good. Okay, so I want to talk about what we call the fourth phase of water. You all know the three phases, solid, liquid, and vapor, and this is something that, if we're right, represents a different phase of water. Now, a lot of you will think that water, that's kind of like the most boring subject on the face of the earth. It's a simple molecule. Everybody knows its properties. It's been studied for hundreds of years, if not more. What else could we possibly learn about water? I mean, do we really understand water? Now, I'm going to start off with a few slides that will demonstrate, I think, that you don't know all there is to know about water. I'm going to show you a few fairly common things, and please think about the explanation that you might give to these phenomena. First one, cloud up in the sky. I saw this today as I was walking on your lovely campus, and the question is, why do clouds float? Now, the clouds are made of little droplets of water. Droplets of water are heavier than air, and so they should come plummeting down, hitting you on the head. How come they float? If you don't know the answer to that, then you really don't understand what there is to know about water. Here's another one. You take two beakers and put them lip to lip, fill them with water, and place two electrodes, one in each one, and impose a high voltage. Turn on the voltage, and here's what happens. Here's what I think happens. I suppose happens, if I could get this video. Oh, there we go. It forms a bridge. And if you move one beaker away from the other, using a mechanical stage, the bridge persists. And it persists indefinitely, and it persists for a distance of up to 4 or 5 centimeters. Please, explain this to me. I don't hear any takers. Okay. Another experiment, this one we've done in our laboratory. You know, if you put droplets of water and let them float on water, the droplets coalesce instantly, right? Well, not in this case. You can see water droplets coming from the tip of this pipette, landing on the water surface, and persisting. And we've seen persistence for as long as half a minute or so. That's not supposed to happen, but it happens, as you can see here. So tell me, please explain it. Okay, I don't see people jumping up. Now, here's another one. This is a lizard. And this lizard is found in Central America, I think Costa Rica. And the video I'm showing you actually comes from National Geographic. And this lizard sits on the limb of a tree. And I don't know, it seems to enjoy itself. And when it gets stimulated in some way, I'm not sure of the stimulation either, it jumps, it jumps into the water. And it walks on the water. Now, because it walks on water, it's referred to as the Jesus Christ lizard. Please explain what's going on. Okay, and this one is my favorite. So this is a container, a trough full of water. And to the water, some red dye is added. I'll explain the significance of the red dye in a moment. It's really for an effect. And sitting behind it is a superconducting magnet, strong magnet. And when the magnet is turned on, what happens is that the water separates and you could walk across the Red Sea with biblical implications. This is real. I saw a demonstration of this. You turn on the magnet and the water separates. So a real phenomenon. Well, anyway, it leads to some problems, especially if you're a fisherman, and if there's no water, it becomes a little difficult to fish. So anyway, I present these to give you some idea that not everything is understood about water. See, and I think you couldn't explain to me about these phenomena, which means you don't really understand everything there is to understand about water, and there's space open for learning some more. Well, I was in a different field. I was in the field of muscle contraction for a couple of decades, in fact. And one day, I had the pleasure at a conference to meet this guy, Gilbert Ling. And I'd heard about him a little bit before, but didn't really know very much. And it turned out he came from China in 1948. It was the first cohort of people, of scientists, young budding scientists to come from China after World War II. There were 4 of them. 3 of them won Nobel Prizes. Gilbert was not one of them. But according to his contributions, I think he should have won at least 2 Nobel Prizes. This fellow was a genius. And so he was also a bit of a revolutionary. He didn't believe everything that everybody else believed. And he said that the water inside the cell was not like this. So we all learn in our textbooks that water everywhere inside the cell, liquid water, And he said, well, not exactly. No, it's not like that. He said, in biology, the water that's inside the cell was not liquid water. It was structured. He used the term structured. What did he mean by structured? He meant that the molecules of water, the H2O molecules, you can treat them kind of like dipoles, a bean with plus at one end, minus at the other end, and these beans would stack upon one another. He called it structured water, like soldiers at attention. And I heard his presentation, and I was deeply impressed. For me, it was mind-blowing because he had so much evidence for this kind of structure inside of biology. And I never heard of it before, but I heard it from this guy. So, I kind of become easily impressed by interesting stuff. So I went home from this conference, which was held in Hungary, and even though my mind was blown by his evidence and other people who presented evidence in support of his point of view, I decided to take one of his books and give them to some of my students. I didn't trust myself. And the students came back and told me uniformly that, if his experiment, if his idea is correct, it changes all of biology. And it looks like his evidence shows that he might well be correct because there's an abundance of evidence. So we had no money to study water. We had money to study muscle contraction. And so well, what can I do? Because I'm entranced by this, and water seems to be even more significant and important than muscle contraction. So I wanted to do something. And what I did first off is I wrote a book. It's called Cells, Gels, and the Engines of Life. And the first half of the book was designed to portray Gilbert Ling's ideas and evidence in a way that's digestible. Gilbert Ling, I think the word editing is maybe missing from the Chinese language. He would just batch something out, send it to the publisher, and it would be published, and it was up to you to figure out what he was really trying to say. So I thought I would do him a favor. It turned out he hated it, and I was kind of surprised, but I think it was that I stole his thunder. And well, whatever, it took years before I could once again curry his favor when I presented evidence that was consistent with his idea. So anyway, the book got differing reviews. At one end of the scale, there was a famous cell biologist from Harvard who wrote, he said, this is a 304-page preface to the future of cell biology. I like that one. And others said, well, everybody knows Gilbert Ling is a crackpot, so pay no attention to this book. It's meaningless. But for for discerning people, they'll choose this book out of any other book that's nearby, as you can see by this. And still, you know, it does take a bit of reading to get the message, as is demonstrated here. So that was the first half of the book. The second half of the book says, well, okay, now what do we do with this? How can you understand some principles in cell biology? You know, if the water is structured, it's kind of ice-like, and nothing can happen in ice, but we know lots of things happen biologically inside the cell. So I was curious about this, and I did a whole bunch of research, and I investigated different kinds of cells, and I found the following as a consistent effect, namely that when the cell was quiescent, that is, for example, in a muscle cell that's not contracting, or a secretory cell that's not secreting, or a nerve cell that's not conducting, the water was indeed similar to what Gilbert Ling was talking about. We found some important differences that I'll tell you about, but by and large, the water structure was organized in some way, and that's shown by these lines that you see here. But when the cell went into activation, became activated, then the water underwent a so-called phase transition that some of you may know about. Physicists and chemists know very well, but biologists less so. A complete change in the structure, that's an all or none kind of change that's initiated, and that's when the cell goes into action. And for a muscle cell, for example, it would mean that the muscle is contracting. So here relaxed, here contracting, and then back again to the relaxed state. And I found that this was true in a host of different cells, with good evidence for this occurring. So not only did this phase transition involve the water undergoing change, but also the proteins. So when the water is in its structured state, it's pressing on the proteins, and the proteins are then in their extended state, shown here on the left. But when the water undergoes transition to the unstructured state, or like liquid water, then the proteins can fold. And it's this folding, from extended to folding, that's responsible for the action of the cell. I could find this in studying a half dozen different cell types, important cell types that you all would know about. So that was the book. Finally, we were able to get some money to begin studying cell biology experimentally. So what's the first thing that we do? Well, first thing that we do, we want to see if we can find additional evidence for this water structure that Gilbert Ling was talking about. Now, what he was talking about looks something like this. So inside the cell, you have various solids, macromolecules, like proteins mostly. And the proteins, their surfaces are mostly, not always, all charged in a way that looks like this. And these are the water molecules. These are the dipoles that I was talking about. So you can imagine, let's say there's a negative charge on the surface here, and this dipole has plus down here, minus here, so it's going to adhere to the surface. And another one here and here, and so because these are adhering, setting a pattern, they tend to structure the ones laterally and also the ones in this direction. Now most physical chemists will agree that there may be one or two molecular layers of structure, but Gilbert was saying, oh no, we're talking hundreds, if not thousands. And I'm going to tell you, share evidence for millions of layers in some circumstance. Now, when this form, this perfect crystalline or liquid crystalline layer, if the water that begins the process starts by having contaminants like molecules, various sorts of molecules, or even particles, they're going to get excluded because you wind up with something that's pure. And so we were looking for a region, this was in an experimental chamber, a region where we started with water and particles, where the particles disappeared from the water. They get excluded. And we immersed, the first experiments, we immersed the gel in the water. And what we found is that around the gel, around every surface of the gel, the microspheres were excluded to a very large extent. So I'll show you a slide. So this is a chamber, and we plunk the gel in, and the chamber contained initially water and particles, little spheres, microspheres. And by the time we were able to get it to the microscope stage, it looked like this. We could see that there was a region right here, in this case 10 micrometers, pretty large by molecular standards, and we watched to see what happened in the microscope. This is one of the first images we got, and we could see that something was building and pushing out the microspheres. And we saw this again and again in not only one particular gel, but many different gels, hydrogels made of water, and also various polymers. And the surface had to be hydrophilic, water-loving. And then this would happen. Someone said, why go through all of this argument about the water that you see here? Why don't you give it a name? So because it excluded, we called it exclusion zone, EZ for short. It works here, Davidson, other places in the US, but it doesn't work in Europe because it's EZ. So it's not EZ to remember, but for us it's EZ to remember. So we call the water in here exclusion zone water. Later, we refer to it as force phase water because every property that we could measure of this water differed from properties of ordinary liquid water. I'll show you a list of some of those properties. So in terms of generality of the phenomenon I just showed you, we found that many, I would say all, hydrophilic surfaces generate exclusion zones, and many solutes are excluded. Obviously, I can't say that all are excluded because we obviously didn't study all, but it seems to be a very general property. So next, is this water really different from ordinary bulk water? I've alluded to the possibility, but I've shown no evidence. So what's the evidence? Well, here's the evidence. This is some of the evidence. Evidence that exclusion zone, EZ water, is physically different from liquid water, bulk water. This is the observation, and this is in red are the methods. And much of this has been published, actually published a decade ago. And if I were to update the list, it would be a lot longer. I'm not going to go through all of these. For example, EZ is more viscous than ordinary liquid water, and the technique we use is falling ball viscometry, et cetera, et cetera. I've got roughly an hour, and talking about 2 or 3 of these would fill up the hour. So I'm not going to bore you with this information, but as I said, it's mostly published. I do want to discuss one feature, which is really important, and that's the third one down. EZ has negative charge, and the technique we use is to directly measure the electrical potential or potential difference of the EZ region and just ordinary water far away. And what we found, to our great surprise, we start with water, which is neutral, and we wind up with EZ water, which is negatively charged. It really screwed us up because how is it possible that you start with something neutral and wind up with something negative? It just doesn't make sense. So we thought, well, there should be a counterpart. There should be a positive charge somewhere else. And we found it. And so this is the scheme. We have, let's say, a gel or some other material that has a hydrophilic surface. And it turns out we start with water, and it turns out after a few minutes that all of this so-called EZ or structured water has negative charge. And we also found, through direct measurement, we found that this region here had positive charge. So what happens is you start with water, which is neutral, and you wind up with a battery. Now, you got negative here and positive here. And battery is interesting for all of biology because you can imagine if you have an electrical battery somehow situated in your cells, you have energy, you have a source of energy. The structure that we were able to deduce, a little more detailed, looks something like this. So here's the hydrophilic material. This starts as just ordinary water. And these layers, these easy layers, which we found have a hexagonal motif, they build one by one, as you can see, just like this. And if you were to look at right angles to the surface, what you see is something like this. So you can see the hexagonal honeycomb motif, and the oxygens are sitting here at the vertices, and the hydrogens are sitting halfway on the linkage between these two. Now, if you take a unit cell and see what you have, it turns out it's not H2O, and it can't be H2O because we found that this region experimentally was negatively charged. H2O is neutral, so it's not. And instead of H2O, if you count in a unit cell, you find that it's actually H3O2. If it were, say, H4O2, it would be twice this. This is neutral, so twice neutral is also neutral. And if you remove one of the positives from it, then you get net negative charge, which is what we measured. So this is the kind of structure that we came up with. So now think about the insides of your cells. The insides are really crowded. This is a gross underestimate. Those of you who have seen, for example, electron micrographs will know that the macromolecular surfaces are very close to one another. Only a little bit of water in between, which means that every water molecule is right near one of the macromolecular surfaces. And these surfaces are charged and mainly hydrophilic, so that the water inside the cell is essentially all easy water. And this is important. And what you learned in biology, if you took a biology course, is that the negative electrical potential of the cell is not due to water, which I'm suggesting it's due to the negatively charged water, but pumps and channels in the membrane. And I've argued in the literature and in the book the many reasons why I believe that argument is erroneous. And I hesitate to go through all the arguments because it take up too much time. But if you, for example, remove the membrane or a large patch of membrane, which constitutes a short circuit, the cell should die, but it doesn't. It persists and it lives. And Gilbert Link, for one, calculated how much energy the cell would need in order to just run the sodium pump. And it turned out to be how many times more, like 30 times?
Yeah, 30 to 50 times.
To 50 times more than the cell could possibly muster. And now we have not only the sodium pump, but we have approximately 1,000 other pumps in the membrane. So if there's not enough energy to run one of these pumps, think about 1,000 of them. But there are other arguments as well. And so another way of interpreting the negative charge in the cell is that easy water is negatively charged, and the cell is filled with easy water. Therefore, the cell has negative charge. And this is, I think, a simpler way. Okay, so we're healthy people. Your cells are filled with easy water. I think most of you are healthy, so most of your cells are filled with easy water. So going back to the answer to the original question, is easy water really physically distinct from bulk water or liquid water? And the answer is yes. It's a layered honeycomb structure that fills our cells. Now, if you have a battery, most batteries, all batteries need charging, right? You forget to plug in your cell phone at night, and in the morning, it's not working. And so the question arises, well, if easy water forms a kind of battery, where does the energy come from to charge that battery? Now, I don't know about you, but we couldn't figure it out. I couldn't figure it out. And then one day, it was an undergraduate student like yourself, a freshman, who was working at a bench just like this, doing the experiment that I illustrated, where we put the gel in and it grew the exclusion zone. He was doing that kind of experiment. And frankly, I don't know whether he was bored doing that or whether he was creative or something. And he saw there was a gooseneck lamp sitting here. So he picked up the gooseneck lamp and shined the gooseneck lamp at the chamber. And the region of the EZ that was illuminated just grew. He called me in, take a look. I was astonished because light, it seemed, light was providing the energy for growing, separating the charge and growing the exclusion zone. You've heard of this before. You've heard of light. In photosynthesis, the first step is light coming in and separating the charges in water, plus and minus. And that step is not so well understood, but it's known to happen. It's the first step. And so what we found is something very similar. So there is precedent. So immediately we repeated his experiment. We confirmed that it was okay. And we started looking to see which wavelength was important, because incandescent light bulb contains an array of wavelengths starting at the short end, ultraviolet and through the visible spectrum, and then at the long wavelength and infrared light. And we found that UV, nothing. Visible light, something, a little bit, and infrared light, huge amount. Our experiments showed that the infrared light for the same magnitude of intensity was 100 times more powerful, for example, than light in the visible spectrum. So now question arises, where on earth do you find infrared light? You know, it seems, infrared light seems a little vague to most people. You know, you look in the toaster, you push down the handle of the toaster, and you see the coils glowing bright orange and giving rise to heat, and you say, oh yeah, that's infrared energy. And that's correct, but infrared energy goes way beyond that. Infrared energy, you can demonstrate that everything in this room is generating infrared energy. So you turn off the projector, turn off the lights, and you whip out your infrared camera, not your cell phone camera. And infrared camera is pretty much the same as another camera, except the sensor is, instead of being sensitive to visible light, it's sensitive to infrared light. So it's completely dark in here, and I could get an image of everything, not only you, but also the chairs that are sitting there, your shoe that's sticking out with your crossed legs, and your cell phone, whatever. So we found that it's incident gradient energy, and this is a real image of a real experiment, but not this obviously. And you can see ordinarily here is the exclusion zone next to the hydrophilic surface, and all the microspheres are beyond here, and this boundary is parallel to the surface of the nucleating material. But if you shine the light, then where it's illuminated, you can see that it grows. And we've seen it grow by, for example, up to 10 times. I mean, it's really astonishing growth. And the most effective is infrared. And infrared, as I mentioned to you, it's all over. So if you were to take an image of this room, as I said to you, everything you can record on infrared camera because it's all generating infrared energy. Now that's the energy that's required for building EZ. See, so if you can imagine a situation, it doesn't need to be in the cell, it could be outside the cell too. Some hydrophilic material sitting next to water, and because the infrared energy is all around, it's there all the time, you'll automatically have some amount of EZ water. And if you increase the amount of infrared energy, then the EZ grows. You take it away, it goes back to where it is. So for this battery, the energy for charging it or for buildup of EZ, it's powered by light, photonic energy, just like the first step in photosynthesis. It charges the water battery, and it orders the water. To create order from chaos, you need energy. That's what the energy does. So now, you can imagine from what I've told you that the situation is pretty straightforward. You want to recharge your batteries, just go lie in the sun. Go to Hawaii or Florida or whatever, lie in the sun and get recharged. Now, if that's true, then you raise the question, if you're the inquiring sort, if you could increase your energy just by absorbing infrared energy, say by lying in the sun, could you capture this energy? Could you exploit it? Could you use it for something? Could the energy be harvested? And what I'm going to tell you now in the next few minutes is the answer is absolutely yes. And I'll give you an example of how that occurs. It all starts with an observation in the laboratory. We found that naphthalene occurs or is manufactured not only in sheets but also in tubular fashion. And I was curious, when you have a tube instead of a sheet, the tube is curved, and I'm wondering, because of the curvature, can you still build an exclusion zone, either outside or inside? So I asked one of the students, a freshman, could you check this out? And the guy seemed to be very competent, and he checked it out, and he had the answer within a couple of days. And the answer was yes, you can find exclusion zones, no problem at all. He was bored, he had nothing else to do. And so I was away somewhere in Europe giving some talks, and it was quite a while ago when doing emailing was a little bit less common, so he didn't bother to tell me about his result and just started doing other experiments. And one day I'm back, and I was telling you the story that I'm sitting in my office and speaking to a colleague who turned out to be important but not so interesting. And I was kind of hoping that the discussion would end. And so this student comes barging in. And usually students don't come barging in. I leave my door ajar and I'll kind of knock softly, are you available? And I'm almost always available. When he comes barging in and he sees I've got a visitor there, he says, I found something interesting I want to tell you. So I pretended that I was a little bit irritated by his barging into my office, but in fact, I really appreciated it because he broke up this not so interesting conversation. So he tells me, I'm looking at this tube sitting at the bottom of the chamber with water around it, and I see the water flowing through the tube indefinitely. It won't stop. And I'm thinking, wait a second, how's that possible? Ordinarily, if you've got a tube in water and you've got flow through it, there has to be a pressure gradient from one end to the other, like in your cardiovascular system, that drives the flow. But there's no pressure gradient here. So I'm thinking, where does the energy come from? And I was thinking, well, we found it's infrared energy that builds the exclusion zone, and maybe that is the driver. And it actually is what we found. So just let me show you how this experiment is done. You take a tube of naphthalene or something else, you fill it with water, make sure there are no air bubbles, immerse it into water plus microspheres, as you see here. Take it to the microscope, and what you see is this. So you've got naphthalene, you've got the exclusion zone here, same here, and it just keeps flowing. We had it going for as much as a day and a half without stopping. So essentially it's indefinite. Well, someone came in and said, well, naphthalene is this weird substance. You ought to try something different and see if you get the same result. Well, another of the undergraduate students did that, and he couldn't find tubes that looked like naphthalene, so he created his own. And so when you have a gel, before it gels, it's a liquid. And as the liquid was gelling, he had this small amount, he stuck a wire in, and when it gelled, he pulled the wire out, and that left a tunnel, an empty tunnel. And he sticks this block of gel with a tunnel into water plus microspheres, and he saw this. So this one is a polyacrylic acid gel. Here's the gel, here's the gel, here's the tunnel, and when he immersed it into water and microspheres, this immediately filled up with microspheres, and as the EZ grew here and here, it pushed the microspheres to the center of the chamber, and if you look at the video, same kind of flow. we tried this with a half dozen different gels and we got essentially the same result. Well, someone said, and we listen to when people come to the lab, someone said, you think this is run by light. Why don't you add more light and see if it goes faster, right? So we cranked up the microscope light, and we found that we could achieve an increase of speed by 5 times, and we published this. So the mechanism that we think is operating here is, so you have a tube, it could be a Nafion tube or something else, and we know that next to the Nafion, EZ grows, negatively charged EZ is here, and the positively charged, the protons sitting in water, hydronium ions, are in the center here. Now, the negatively charged EZ is basically stuck to the surface, but the protons, or hydronium ions, are free. They start repelling each other as they build up, and all they want to do is escape. So they can escape from this end or the other end, and that's what's shown here. So we can measure the protons actually escaping, or the hydronium ions, that has protons stuck to water. And as they escape, new water comes in to feed the inside of the tube. And the process is perpetuated, and it keeps going. And the basis for it, essentially, is the EZ phenomenon, and that's powered by infrared light. Okay, so please bear that in mind for a moment. We have basically a hollow tube. Work is done by pushing the fluid through because it has viscosity, so you need energy to drive it through. Energy is required, and the only energy that we could conceive of in a case like this is the infrared energy absorption from outside as essentially the power source. So we conclude from this experiment that water transduces light energy. It's a transducer, converts light energy into mechanical energy from this kind of experiment. Now, so the question arose, does human biology use radiant energy or infrared energy? Now, suppose you're Mother Nature, you know, and you've done pretty well with green plants and some single-celled organisms using light, and you decide, hey, you know, I'm bored, I think I'll create animals. Now, animals can forage and find their own source of nutrients and food, so you're stuck with a dilemma. Should you throw away this light-mediated energy production, or keep it, maybe in reserve, or in addition to getting energy from other sources? And I think, I'm not sure, but I think most of you, being bright young undergraduate, you think, well, you know, if I were Mother Nature, I'd keep it in reserve. Why the hell not? You know, why throw away something successful? And so that philosophically, that, you know, it might be that we actually use light to get energy for our body. And the first thing we began thinking of was maybe, you know, we get radiant energy all the time, and some of the wavelengths can penetrate pretty deeply. And we thought maybe the cardiovascular system, you've got capillaries right near the surface, and if infrared somehow comes in, it could conceivably feed the capillaries in some vague, unspecified way, and give us energy. So now I didn't think seriously about this until I went to Russia. I met my friend, Professor Vladimir Bayakov, who was at Moscow University. And of course, we had some pleasantries, a bit of caviar and some vodka and stuff like that. And he said, we got to go to the laboratory. So I went to the laboratory with him, and he wanted badly to introduce me to his colleague down the hall. And he introduced me, and the colleague comes in, and he says, there's a big problem in the cardiovascular system. And, you know, I'm reacting, oh, come on, tell me your big problem. I must admit to a measure of arrogance, because I had studied cardiovascular fluid dynamics as a graduate student, and I thought we had it kind of all worked out, and this guy is telling me there's a big problem. So what could the big problem be? You know, I was waiting to hear blah, 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 and yawn. He had me convinced in 5 minutes that there was really a problem in the cardiovascular system. Shame on me. So what's the problem? Well, he said, here's the problem. red blood cells have to pass through the cardiovascular system, including the capillaries. Red blood cells are 5 or 6 micrometers in diameter, but the capillaries, many of them are as small as 3 or 4 micrometers in diameter. So there's a size difference, almost a factor of 2. So the red blood cells that we've all seen pictures of, you know, it's like donut shape, it's filled in the center, they got to bend. And we saw evidence for this kind of bending, which you can see here. So this is muscle tissue, and this is a capillary, and this is a capillary, and even this is a capillary. And you can see as the red blood cells pass through, they get squeezed. So they don't really look like this, they look like that as they pass through. Something has got to squeeze them. So what's squeezing them? Well, he said, well, one possibility is that as the ventricle builds pressure, it's enough to squeeze them. He calculated it's impossible. He said, according to his calculations, the pressure in the ventricle would need to be something like 1 million times the pressure that actually is generated in the ventricle in order to produce the energy to do the squeezing. He said, there's got to be something else. And even if his calculations are off by a few orders of magnitude, we're still thinking of very, very, very high blood pressure to do it, which doesn't exist at all. So he said, there's something else. So he began telling me about all the possibilities that he's thinking of. There were like a half dozen, including bubbles in the blood and a few others. And meanwhile, I'm thinking, we have a finding in the laboratory that could be relevant here. We have a finding that shows that you take a hydrophilic tube, put it in water, and the flow goes through, built on infrared energy and the buildup of EZ. And I'm wondering, is it possible that our finding applies to the cardiovascular system, that what's really going on is it's not just the heart that's contracting, but also the vessels themselves are also pumping the blood via infrared energy in a way that we found in the laboratory. In other words, that maybe radiant energy is driving the flow in the vessels, that is, over and above your heart. And so that's the question that arose. And as I was describing this morning, I asked one of my students to check this out. And he told me, after he got a positive result, which I'll show you, he said, you know, when you proposed this research area to me, I thought you were on some kind of drug. I couldn't imagine that it's the vessels also that are propelling the blood, not just the heart. So he did the experiment, and he did it in a 3-day-old chick embryo. And chick embryo, 3 days, it has a pretty well-developed cardiovascular system, but the regulatory systems, the hormonal and neural, are not well developed. So it's a pretty pure system. And what he did was developed an optical technique where after he lopped off the top of the fertilized egg, he could image a flow in any of a number of vessels. Okay, so what did he do? Well, he tested the signature feature of the mechanism that we found in the lab. That is, it's run by infrared energy. So here's his result published about 1 year ago. This is blood flow on this axis and time. So first thing that happens is he injected into the heart, he injected potassium chloride, and that stops the heart. Now, if you stop the heart, you expect the flow to stop, but the flow didn't stop. The flow went down to a low level and persisted. It didn't go down to 0. And then he tested the signature feature, which is to add infrared. And you can see after he added infrared, the flow increased by roughly a factor of 3. And then he took away the infrared, and it went back down again. So reversible. So it doesn't prove that the needed mechanism beyond the heart is what we found in the lab, but it's highly suggestive that that's the case, because the prediction about infrared was followed. We since found, or he did a thorough literature search, and he found a half dozen papers published over the past century, a half dozen papers that demonstrated in completely different contexts that if you stop the heart, the flow doesn't stop. So, you know, it doesn't take a rocket scientist to figure out that there must be something besides the heart. And we think we've uncovered what that something is, this principle that we found in the laboratory appears to apply in the heart, which basically means that you folks, maybe me too, it's not just the heart that's pumping the blood, it's the vessels also. We don't know yet, technical issues prevent us from figuring out what fraction of the energy is from the heart and what fraction is from the vessels. But if we're right in this, it means that in your cardiovascular system, it's not just the heart, also the vessels that are operative, which of course has important implications and suggests that indeed the energy from EZ is really operating in your body. And there are more examples, I just don't have time. So now it doesn't mean that you're photosynthesizing like the plants, but it does mean that you're somehow using light energy to drive processes inside your body, which you don't ordinarily think about. Okay, light supplies energy. Now, so for building energy and creating function, should all of this matter to you? Well, light matters and water matters, maybe more than you might think. So the question is, what builds EZ water in your cells? Because to keep healthy, you need this EZ water, and you want to know what is there. And so I'm going to list a half dozen. First of all, water is the raw material for building EZ water. So if I drink this, some of this water gets converted to EZ water. So keep well hydrated, and everybody knows that they have to drink a goodly amount of water. The second one is green juicing. I know some of you know about this, others don't. And what is all of this about? So you go into your backyard, you see some nice plants that are growing, you rip off the leaves, you put the leaves through a kind of crushing machine and squeeze out the water and drink it. What is this water? Well, it's the water from the inside of the plant cells. And these are freshly grown plants, and just like our cells, these plants contain a lot of EZ water. So you're basically drinking EZ water. And that's one way of replenishing what might be missing from your body. So, and this is touted by many health practitioners as the simplest thing that you can do to improve your health. Substances like turmeric, coconut water, etc., they're known for ages, for 5,000 years from Ayurvedic times. They're known to promote good health. And we wondered about that, and what we found is that these substances, every one of them that we tested, builds EZ water. And so, possible reason why they're so effective when you've got some illness, and you take these, is it rebuilds EZ water that may be missing from your cells. We try to poison, the opposite, glyphosate, which is the weed killer. And some people say, well, it doesn't only kill weeds, it kills us too. And we saw only a diminution at every concentration of EZ size. So the opposite finding. In Seattle, in the wintertime, we don't see the sun a whole lot. It's gray. And unlike the weather here, it's not so pleasant when tourists come to Seattle, and they come in the summertime. In the wintertime, you know, if the sun happens to poke its head out, you see smiles on people's faces, and you wonder, you know, what is it? Some psychological effect? It may well be. But also, when the sun comes out, it's generating a lot of infrared energy. So we receive suddenly a burst of infrared energy, roughly half of the sun's energy. We receive this, and as I tried to demonstrate to you, it builds EZ water. Buildup of EZ water, good for our health, and I think good for our mood too. Just gets into our brain cells, and the infrared energy, some of the wavelengths do pass through the skull. There's imaging that's based on external source of infrared, goes through the skull, gets scattered around, comes back out again, and you get an image of the brain. So demonstrably, it passes through. And I think that that could be the explanation for that. Sauna, well, it's heat, and infrared energy is the source of heat. So you're basically feeding yourself infrared energy. And I know from speaking to you, many of you have had the experience, go into the sauna, come out and feel a whole lot better. If you're feeling depressed, you feel undepressed. If you got pain in your rear end, you don't have pain in your rear end. If you're tired, you're awake when you come out, et cetera, et cetera. So I think the mechanism is a build-up of easy water. And then grounding, or so-called earthing. You connect yourself to the earth. Now, I started my career studying electrical engineering. And we were, of course, taught about grounding, or the British or the Europeans say earthing. And no professor ever even hinted to me or anybody else that the earth might be negatively charged. But in fact, the earth is negatively charged. And you probably don't know about it because nobody ever taught it to you, but I understand that in Russia, people of my generation, every middle school student knew that the earth was negatively charged. But they don't teach it here. I'm sure they don't teach it at Davidson College, or maybe I'm wrong. I never learned it even as a student in electrical engineering. Nobody even hinted that that could be the case. And when a Russian guy told me that the earth is negatively charged, and there's actually a potential difference between the earth and the atmosphere above, I went home and I couldn't sleep that night because this was shattering. Next morning, one of my students came and brought Feynman's lectures. And some of you may know Richard Feynman, the great Nobel laureate, and every physics graduate student reads his lectures. Volume 2, Chapter 9, the Earth's negative charge, and a whole chapter based on the evidence for that. So 50 years ago, it was well known. Right now, nobody knows about it. That doesn't detract from the truth of it. So the Earth is negatively charged. Now, what does that mean? Well, it means that if I walk on the Earth right now, I've got insulating soles on my Birkenstocks, and nothing comes through to me. However, if I take off my shoes and socks and walk on the wet grass, I'm electrically connected to the Earth, which means that the negative charge from the Earth has the capacity to be infused into my body. And cells that don't have enough EZ don't have enough negative charge. And so the negative charge from the Earth can replenish that. And we have direct evidence. If you take ordinary water, put negative charge in, it converts to EZ water. So I think that's what happens when you ground yourself. You can do that. You can do that by walking on wet sand right next to the ocean, or walking on wet grass, or immersing yourself in a mud bath, or even swimming in the ocean. All of the above. And that will restore your health through a very simple means of building easy water. I want to end with a few slides about cancer. We got into thinking about cancer because all this stuff makes you think about stuff, and all of us have been touched by the scourge of cancer, one way or another, either personally or family or friends or what have you. And so, first question to ask, cancer, the cells keep dividing. So, why do cells keep dividing? And this question was asked 50 years ago. What prompts cells to divide? And it was asked by this guy, Clarence Cohn, early 1970s. And he came to the conclusion in his studies that it has to do with low magnitude electrical potential. So now, a typical cell in your body has an electrical potential of minus 50 to 100 millivolts, magnitude 50 to 100. And it turns out that this guy, Clarence Cohn, measured, he took cells, and I think it was not originally cancer cells, but other kinds of cells, and he found that if the magnitude of the potential was small, the cell kept dividing, like minus 10 or 15 millivolts instead of minus 50 to 100. The cell kept dividing. And he took measures to increase the cell potential to minus 150, the cell would never divide. So he thought the electrical potential was critical for initiating cell division. Well, recently I looked for a review article that dealt with this issue of cell potential and division. And here's what I found. So it looks a little complicated, but it's not really. So this is the so-called membrane potential, which I mean cell electrical potential. And here's 0, very small, and a little bit bigger, magnitude, and so on. And here, at this end of the scale, is minus 90 millivolts, a very robust electrical potential. And these reviewers here were looking not only at tumor cells that divide, but also non-tumor cells like embryonic cells that naturally divide. And so he called them proliferating cells and non-proliferating. And this is a review article. So what you can see here is that if you've got tumor cells that are dividing, or non-tumor cells that divide, the electrical potential is small. And if you look at cells that are non-proliferating, that is non-dividing, you can see the electrical potential has a large magnitude. So the point is, if you have a small electrical potential, the cells keep dividing. If you have a larger magnitude potential, they don't divide. Now, lots of studies, this is not all that was published, lots of studies show small electrical potential dividing. Now, so what about small, what does small electrical potential mean? Well, in terms of what I've been telling you, small electrical potential means not much easy water. So tumor cells, cancer cells, have not much easy water. And the reason they don't have much easy water is that if you look at the structure of these tumor cells, they're kind of empty. And all the surfaces that are needed to nucleate the growth of easy water are absent. So you have small electrical potential, and the small electrical potential causes the cells to divide, so they keep dividing. And if this is true, and I have a paper out on this subject and this suggestion, if it's true, then a therapeutic approach to cancer is build easy water, just by those measures that I listed, half dozen different measures, and there are more. And surprisingly, there's a good deal of evidence that some of these really have an effect, already shown. So I think this is a new approach to cancer that's based less on genomics and more on functional aspects that we know about. So, the same approaches that I described a moment ago should be useful for treating cancer, reversing cancer. So healthy people are filled with, cells are filled with easy water. If you don't have enough easy water, your cells are dysfunctional or, at the extreme, cancerous. I end with practical applications beyond health that come from our studies. And one of them is getting energy from water and sunlight. So you've seen this before, and the idea is you put one electrode in here and one electrode in here, and negative and positive. You measure a potential difference, and if you connect it to a load, like a lamp or LED, a lamp or something, should be able to light it. And here's the experiment that shows that it works. So on the right side, you have, I think, a dozen or so chambers, and in each chamber, you have a Nafion surface, a negative EZ, positive zone beyond. You put two electrodes in, one negative, one positive, and you connect these cells either in series or parallel, depending on whether you want a lot of current or a lot of voltage. And this is a switch that connects it all. And there's an LED lamp here and a magnifier, and you can see what happens. This is proof of principle that you really can get electrical energy from water. I think you haven't thought about that option before. And of course, we're interested in building upon this proof of principle to see if you could scale this up to light a house or even a city. It might be, and if we can achieve that, then this is a kind of technological revolution that comes from that. We don't know if we can, but it remains to be tested. Another one is getting drinking water from contaminated water. So you put input water in, which may have pharmaceuticals in it or microplastics or something, and then you take a Nafion tube. It doesn't have to be Nafion, but this is representative. And remember what happens in a tube like this. You get EZ here, and all the junk, the contaminants, are pushed to the center. And you put here what we call the fancy name differential extractor, but it's nothing more than two concentric tubes. And the center tube picks up the junk and dumps it, and the outer tube is actually EZ water, which is pretty much junk-free, and you collect that also. So, so this is a filter. There's no physical filter. We call it a filterless filter, no filter to change. It's separated by virtue, well, ultimate infrared energy from the environment. So it's a simple device, and we spent a few years perfecting it. It looks promising. Now, the real gangbuster effect is if we can separate salt. So the idea now, and we have some preliminary evidence, not a whole lot. We treat the contaminant as salt. You start with ocean water, which got a lot of salt on it, in it. And if the salt really separates to the center, as we think it does, then the same principle can be used to separate the salt, which is here, from the purer or pure water, which is here. And we don't know for sure if we can do it and make it happen, but if we can make it happen, it means that you can basically drink ocean water. And if this works, again, needs development, then we have evidence that we can do something wondrous for our lives. So in conclusion, I guess the center point of all of this is that water actually has 4 phases, not 3. And we know about the 3, vapor, liquid, and ice. And we found evidence for a 4th phase of water, which has kind of liquid crystalline properties. And we also found, no time to show it, if you want to freeze water into ice, you must go through this state. And if you want to melt the ice to water, you must go through this state. It turns out that it makes understanding a lot easier if this is the case. So we have evidence for that. So that's why this easy water is put between these two. And all of this has broad implications, many implications. And I guess perhaps the most central one is that water is absorbing radiant energy, infrared energy. Water is absorbing from everywhere. That's sort of central. And I've demonstrated to you that one of, among at least several biological effects in blood flow, in driving blood flow. In chemistry, if you read the chemistry book, you'll find none of the features that I've been describing. The effect of radiant energy, the separation of charge, any of that. And so if what I'm suggesting to you is true, if it's true, many of the interpretations of what happens in chemistry and in aqueous chemical reactions, the interpretations will change. Hopefully get simpler, because some of them are pretty complicated. Another one is weather. So you listen to the weather forecast. Here it's perennially nice. In Seattle, it's not. So we see, in the wintertime, we see clouds, and it's not, the forecast is not always very helpful. 50% chance of rain. So should we take our raincoat, or should we leave it home when we go to work? It's not 50% probability. It's not so helpful. And so the forecasts are, not so great, not so good, reasonably good, and they're based on historical trends. So what is the environment like previous cases? And probably this is going to be the same. So that's where you get 50% probability or 60% or whatever. Well, it turns out that clouds are charged. Now, you know that thunderclouds are charged because you see evidence of it, but in fact, evidence, again, from 50, 60, 70 years ago, show that all clouds are charged, even the ones that don't unleash lightning and thunder. I'm not sure if it's true of you, but I've never heard the weather forecasters talk about the charge in clouds. Yet, I think the charge in the clouds is one of the most fundamental attributes of the clouds. For example, the reason that the cloud floats is it's full of negative charge, and the Earth is full of negative charge, so the two are repelling each other. Very simple explanation. And so I think that if you understand how clouds, how even you can generate a cloud, because that's not so obvious. I treat these in the book that's coming out in a couple of months called Charged. And I got 4 chapters on weather and how weather works, starting from first principles. If I challenge you to find out how a hurricane occurs with a characteristic twist and such, starting from first principles, you'd be pressed to do it, because I don't think it exists. I've tried to do it, and charge is absolutely critical to that. So I think, if what we're suggesting has some merit to it, we'll soon be able to understand the genesis of weather. Health, well, I've alluded to the fact that easy water is absolutely critical. You need to have a full complement of easy water in order for your cells to be healthy. When you get to be my age, wrinkles appear. Wrinkles are a sign of dehydration. And also, when you get to be my age, you get various afflictions, more than you younger people get those afflictions, because you don't have enough easy water. You're dehydrated. And dehydration is capital crime. If you want to freeze food or dehydrate food, it's nice to know something about the principles of how they work. And in terms of practical applications, so-called practical filtration, filterless filter is one application. And an application of that application could be desalination. If it works, remains to be seen, remains to be developed. And also getting electricity from water. Who could imagine that you could get electricity from water and infrared energy? But we have proof of principle. So all of, or much of this appears in this book, the 4th phase of water, which turns out, if you look on Amazon, it's very, very popular. And the next book that's coming out, I don't have a picture yet, but I should have one. It's called Charged, and that deals with a lot of things in the environment that we think we understand, but we don't really understand them, including not only weather, but also gravitation, how birds fly, how planes fly, how fish swim, and a whole lot of other things. So I will end there, and I can entertain, if we're okay on time, a couple of questions. Thank you very much.